Welcome, class. It's Dr. Lindner, and in this video, we're going to be talking about the different classifications of tissue. And there are four classifications that you can see. We have epithelial or epithelia, connective tissue, muscle tissue, and nerve tissue. So remember how these tissues form. Tissues are only created because a group of cells come together, they join by those cell junctions, whether they're desmosomes or hemidesmosomes or gap junctions or tight junctions. They hold cells together. And when cells come together of similar structure and function, it's going to create these different types of tissues: epithelium, connective tissue, nerve tissue, and muscle tissue. Now, when we talk about tissues, let's just focus on these first two for a second, epithelium and connective tissue, because number three and number four, I think, are the easiest to go over and the easiest to learn. One and two have a lot of subcategories to them. And what we need to be able to do is to differentiate epithelial tissue from connective tissue under a microscope. So when we look under a microscope, we may see a bunch of cells with a nucleus, and they may be very closely packed together, like we see here. And when we see cells that are closely packed together with very little extracellular matrix, extracellular matrix. Sometimes we'll abbreviate that as ECM, extracellular matrix. Extracellular and intracellular, we know they mean different things. Intracellular means within the cell, extracellular means outside of the cell. So when we have extracellular matrix, which is the cement that's between these cells, if we have little extracellular matrix and the cells are closely packed together, well, that's going to be epithelium. But if we have some cells that are not so close together with some protein fibers and another cell with a different type of nucleus and then another cell with a different type of nucleus, when we see cells that are far apart from one another with increased or more extracellular matrix, okay, now we're talking about connective tissue. So there's a big difference in the way these cells are arranged further apart from one another than the way these are very closely packed together. So that's like the first type of question you would ask yourself if you ever were asked to determine what type of tissue is this, epithelium or connective. You go, okay, are the cells closely packed together or are they not? If they're closely packed together, it means they have little extracellular matrix. That's going to be epithelium. If it's made up of a bunch of cells that are far apart from one another, not closely packed, and they have lots of extracellular matrix, that's going to be connective tissue. Okay? Keep that in mind. Now, epithelium is a lining and a covering type of tissue. So when we think of a covering, I think of the skin, the dermis. So when we have epithelium that covers the external surface of the body, the upper part of the skin, remember upper is epidermis, the epidermis is made up of epithelial tissue. Epithelium can also be a lining. It can line body cavities and tubules. What do we mean by tubules? In the word tubule is tube. So anything that's shaped like this. It could be like that. It could be like that. Right? That's a tube. Tubules. So tubules are anything that looks like a hose. So maybe your blood vessels, maybe your esophagus, maybe your trachea, maybe your small intestine or large intestine, anything that's tubular. When we get into the kidneys are made up of lots of tubules. In order to detoxify and make urine, it has to go through the kidney tubules. Glands. Glands are classified as epithelium. So we have sweat glands, we have oil glands, we have mammary glands, we have earwax glands. So there's different types of glands throughout the body. That's lining, covering, and glands. Now we're still looking at epithelium. We're only focusing on epithelium here. And we have to figure out how to name epithelium. So remember, the first question you asked yourself, was, do I have a bunch of cells that are closely packed together with very little extracellular matrix? Which was this. Bunch of cells closely packed together with very little extracellular matrix. If that's what it looks like under a microscope, it's epithelium. But now we have to name the epithelium. So the second question we're going to ask is, how many layers of cells do we see? Do we see one cell layer, or do we see more than one cell layer? If we see one cell layer, then we refer to that as simple epithelium. That would be simple right there. And if we see more than one layer, then we're going to call that stratified epithelium. Okay? So the second question you would ask, now that I'm looking at it under a microscope, how many layers of cells do I see? Is it one layer or multiple? And it's the number of layers that you see either at the apex. So let's say we're looking at a slide. It's whatever we would see up here at the top. Or if you're looking at a tubule, a cross section of a tubule like this, it would be the layer that's just next to the lumen. So the lumen is the hole. So being that we have one cell layer around that lumen, that would be simple epithelium. Now, if it would look like this, I'm making another layer. Now that would be stratified epithelium. Okay? Now that's the second question, but there's three questions. The third question is, what's the shape of the cell? Is it the ones that I've been drawing here that look like dice or look like cubes? That would be cuboidal. Do they look like tall buildings? That would be like tall columns. That would be columnar. Or do they have this very, very flat, irregular shaped appearance? And if it has that irregular shaped appearance, then we call it squamous. So if we were to look here, we could see these are flat. These look like ice cubes. And these look like tall columns. So if we see that they're one cell layer thick, it's simple. If we see that there are multiple layers, it's stratified. Simple, squamous epithelium or stratified squamous epithelium. Here, it's one cell layers of these cubes, so it's simple cuboidal epithelium. Here, it's more than one layer of these cubes, so it's stratified cuboidal epithelium. Here, these are tall buildings or columns, and they're one cell layer. See how the nuclei are all even? That's how you can tell how many layers there are. So here, there's two layers. Here, there's one layer. Here, the nuclei are all aligned, one cell layer, so it's simple columnar epithelium. Here, we have multiple layers, but notice the nuclei are nice and aligned, so we call it stratified columnar epithelium. Now, there's another type of columnar that we see on the bottom right, and it's called pseudo-stratified. The word pseudo means false, so it's falsely stratified. It looks like it's many layers because look at the nuclei. The nuclei are at different levels. And because the nuclei are at different levels, it gives it a stratified appearance, but it's only one cell layer thick. So it's pseudo-stratified columnar. And usually where we have pseudo-stratified, we have something else right here, and those are called cilia. These little brush hair-like structures here. And these beat rhythmically. They're beating rhythmically. So here's a tubular system. And let's say this is the trachea, your windpipe. And we're going to make columnar cells here. How about that? We make a bunch of columnar cells, tall columns. Let's put nuclei in there. And let's add some cilia. Those little hairs right here. And these little hairs are beating rhythmically. They're designed to beat upward because when you breathe in air, right, oxygen is coming down into your lungs. So all the debris is going to stick to these cilia because we have an interesting type of cell. We'll make it green. We'll make it green because green starts with G, and those green cells are called goblet cells. And what goblet cells do is they make mucus. So these goblet cells are going to secrete mucus to the surface, and you're going to have mucus that sticks to the top of these cilia. But remember, the cilia are beating upward, so any type of dust or particles of debris that stick to this glue, to this mucus, it's going to prevent it from going deep in the lungs. It's going to push it upwards. How cool is that? The problem is with smokers. When people smoke, they burn off the cilia. And when they burn off the cilia, you have nothing to trap the debris. So all the carcinogens from the smoke get deep into the lungs. And when these type of cells die, when these columnar cells die, they're replaced by a different type of cell called a squamous cell. When those ciliated columnar cells die, they're going to be replaced by this, squamous cells. And when one cell type is replaced by another, there's a term for that called metaplasia. Metaplasia means one cell type has died and it's morphing and becoming something else that it shouldn't. That's metaplasia. Women know this pretty well because when women go to the gynecologist, they'll do a pap smear and they'll talk about a type of metaplasia, but they'll say that the woman has cervical dysplasia, where the cells are altering and changing shape. And typically dysplasia or metaplasia can be precancerous. So they monitor that and watch that. When these are burned off from smoking, all the debris makes it down into the lungs, to the deep part of the lung. And that's why when that smoker goes to sleep and they wake up in the morning, they have the smoker's cough. They're like, right? They're coughing so hard to try and get all the debris that made it into the lungs. They're trying to get it upward because the beating cilia that typically beat upward are no longer there. Okay? So that's the cilia, those long, slender extensions, and they help to move the secretions and the fluids across the surface membrane. And if they're smaller than cilia, they're called microvilli. And microvilli are used to increase absorption. When I think of microvilli, I think of a sponge. And there's lots of microvilli. If, let's say, this is another tube, let's say it's the intestines, we're going to have columnar cells here, and there's going to be lots of cilia, and even smaller than that, microvilli. And all the nutrients, the carbohydrates, the proteins, the fats, they're going to be absorbed by those microvilli. And then here, inside these, there's going to be arteries, there's going to be blood vessels. So the nutrients make it into these blood vessels. They're absorbed by the microvilli into the blood vessel, and then it brings all the nutrients into your circulation and bring all the vitamins and minerals and carbohydrates and proteins for all the other parts of the body. So we need those microvilli. That's why when people have, let's say, celiac disease, or they have issues with gluten, these microvilli break down, and there's a lot of malabsorption diseases because these people can't absorb nutrients very well. Okay. Okay, so when we look at the epithelium, let's just take a look again. We look at it from the side view. We see that these are the flat ones, and it's only one cell layer thick, so it's simple squamous epithelium. These are simple cuboidal epithelium, and these are simple columnar epithelium, right? Cube-shaped, column-shaped, and flat-shaped, and they're all one cell layer thick. Down below, these are stratified. We go to the top, not the bottom. We go to the apical surface, the top part, and we see, okay, we got a few layers of these flat, irregular shaped cells, so it's stratified squamous. Here we've got 2 layers of the cubes, so stratified cuboidal. This one looks like it's stratified because the nuclei are at multiple levels, so it's pseudo-stratified columnar. Now there's one that I left out, it's transitional. I want you to think of a balloon. If, let's say, this is a balloon and it's deflated, and we draw with a marker a square on that. But then you blow air inside the balloon and this starts to inflate. What happens to the shape of that square? It starts to change shape. It transitions. It can look rectangular, it can look round, right? It alters in shape. So whenever we have parts of the body where the organs can inflate or deflate, we're going to have transitional cells. So anything that has to do with the urinary tract, especially the urinary bladder, right? If the urinary bladder is filled with urine or distended or not, those cells are going to alter in shape. Okay, so this has shown up before on some quizzes with the McGraw-Hill Connect, and it's a drag and drop, right? So we have simple squamous, simple cuboidal, stratified squamous, pseudostratified columnar, and transitional. And they want you to kind of have an idea as to where do we find it or maybe what their functions are. Now, I want you to understand that if something is simple, let's say it's one cell layer thick, it's easier for things to pass through that one layer compared to if it's multiple layers, right? If it's multiple layers, maybe it's for protection, but if it's one cell layer, things can diffuse or pass through in both directions. Maybe oxygen is trying to get in or carbon dioxide is trying to get out, okay? If something is stratified, if it's multiple layers, we think of protection primarily. Okay, so now the epithelium can function in diffusion, filtration, secretion, and protection based on location. That key word there is diffusion. That's going to be simple squamous epithelium. Simple squamous. So that means you're going to see this where we have the lungs, deep in the alveoli, where we're going to have the gas exchange. Simple squamous epithelium. Simple cuboidal, these are for absorption and secretion. We're going to see these where we have the kidney tubules. There's something called the proximal convoluted tubule and a distal convoluted tubule. Proximal convoluted tubules and distal convoluted tubules are part of deep microscopic part of the kidney, a specific region called a nephron, and it's going to be lined with simple cuboidal for absorption and secretion. Stratified squamous, right? Now you're going to have flat, irregular shaped cells, but these are going to be multiple layers. So that's going to be the epidermis for protection, skin, that upper layer. Pseudostratified, remember pseudostratified is when you have those columnar and you have those cilia, but the nuclei are at different levels. So it gives the appearance that it's stratified when it's not. And remember with this, you're going to have those goblet cells located in there. And remember those goblet cells do what? They secrete mucus that stick to the top of the cilia. So pseudostratified columnar epithelium functions in the movement of a secretion, like mucus, because of those goblet cells, across the apical surface. Pretty cool, just like the trachea. And transitional, remember the balloon, blowing up the balloon, accommodates distension and relaxation with changes in the organ volume, the urinary bladder. Okay, so let's take a look at where we could find some of these. Now we're looking at a picture of this. So here's the pseudostratified, right? Look at these. These are column shaped, but notice the nuclei are scattered at different levels. So it's pseudo stratified. And way up here at the top, you can see those little hairs. So it's pseudostratified ciliated columnar epithelium. And these are found here in the trachea, in the respiratory tract. Remember, those cilia are going to be beating rhythmically upward to push debris out. So we'll find them in the respiratory tract. Let's look down here in the lower right, stratified squamous. So this is the bottom, this is the top. And when we look at the apical portion, the top portion, it's multiple layers of the flat irregular cell, so it's stratified squamous epithelium for protection. You'll also see it of the skin. Why? Protection. Maybe down the esophagus, maybe you swallowed a pit, maybe a cracker with a sharp corner to it, and it scratches the esophagus. So we need multiple layers. Imagine you only had one layer of squamous cells on your skin, and you felt something crawling on your skin and you scratched it with your nail. you'd slough off that one layer of squamous cells so easily. But when you have 25 to 30 layers of this, you could scratch it many times. You're not going to be bleeding. Okay. Simple columnar epithelium. These nuclei are at one level. These are column shaped. We'll find these in the intestine. And you may have little hairs here as well. You can have cilia as well. In the kidney, remember the kidney tubules, whether it's the proximal convoluted tubule or the distal convoluted tubule. Remember I said epithelium, we either look at it at the apex, what's at the top, or we look at the lumen, the hole, and we see what type of cells are adjacent to the lumen. These are cube shaped, but it's only one cell layer, so it's simple cuboidal. And here we're looking at the lungs, the alveoli, or the air sacs of the lungs, where it's simple squamous epithelium. It's only one cell layer, so it'll allow oxygen to go through or diffuse, and carbon dioxide to get out of it. Okay, and this is kind of really important stuff because understanding epithelium helps us understand a lot of health challenges that people have, because when epithelial tissue degenerates, we can have problems. So, for example, let's say in the intestine, right here, let's say the person drinks alcohol, or they take antibiotics, or NSAIDs, non-steroidal anti-inflammatory drugs, or they use what do they take for inflammation? When they have chronic inflammation, they can take a lot of powerful pain meds that damage the intestines. But what it does, these cells are held together by what's called tight junctions. And if, whether it's high sugar, fried foods, antibiotics, alcohol, drugs, prednisone, anything like that, it weakens the tight junctions. So now, instead of these two cells being held closely together, now you'll have a cell like this and another cell like this with a gap between it. Now remember, your blood vessels are here and nutrients are trying to get through to get absorbed by the blood vessels. Now, if you have your intestine and it's breaking down your food, the carbohydrates, the proteins, the fats, and it's breaking them down into small, small bits, these cilia can absorb them as they're supposed to, pull them into circulation. But if the tight junctions are damaged, you can have proteins that escape and slide through larger than what they should be, get into circulation, and guess what your body does if a protein invades your system and it doesn't recognize it? Your immune system attacks it, and you build up antibodies to this type of protein and anything that resembles it. And they call that leaky gut syndrome or intestinal permeability. So if this happens to be the protein in eggs and it matches the protein on the outside of the thyroid, there's something called molecular mimicry where your immune system makes antibodies that just don't attack this protein, but any protein that resembles it. And if the protein that resembles it is on the thyroid, you attack the thyroid as well. And you can get a Hashimoto's. Or if it matches the protein in the joints, you get a rheumatoid arthritis. So there's lots of new cutting edge research on this intestinal permeability and a lot of autoimmunity. Intestinal permeability. Good place to take our first break. When we come back, we'll get into connective tissue.